Okay, welcome to our genetics unit. Wait a minute, we just studied DNA, right? Aren't we done with genetics? Well, we would be, except for that what we're going to study now is how that DNA that we studied in the molecular form, how that DNA actually expresses itself when we see the traits in an actual living thing. And we're going to look at how those traits get passed on from one generation to the next to the next. And this is based on our entire study is based on the work of essentially one man, the father of genetics, is Gregor Mendel. So we're going to first take a look at what Mendel did, and then we're going to take a look at how we apply what he learned to how we study genetics today. He gave us pretty much everything we really needed to know. Obviously, we've learned a whole lot since Mendel did his work, but he really gave us all the fundamental laws of genetics that we're still using to this day. Okay, so without further ado, we're going to go ahead and move on. So let's get some basic definitions out of the way. Genetics is the study of heredity. So what's heredity? Heredity is the passing of traits from one generation to the next. Serious study of how traits get passed from one generation to the next began in the mid-1800s, but the ideas and the studies done back then weren't all that great. The main idea that was popular about heredity in the 1800s was an idea called blending, which was essentially like taking two colors of paint, red and white, you mix them, you get pink. Well, you get a mom and a dad, you mix them together, you get a blended offspring. Obviously, that should have been pretty obviously a wrong idea to most people. There are just some traits that aren't blended, okay? Your parents are a male and a female, and you are not a blend of them. You are either one or the other. There was really no answer for that, and there's lots of other traits that don't appear to be blended either. But people back then just really didn't have an answer for that question. And that could be because their experiments were designed so badly. For example, a genetic study during that time might have been, how is beauty passed from one generation to the next? Well, that's just a really wretched experimental design right from the get-go. Obviously, beauty is horribly subjective, and many of the experiments at this time were like that. They didn't look at one specific trait, but they tended to look at just large qualities of people, of intelligence, or various things that are really a bunch of traits working together. They didn't isolate it to just one trait. So additionally, most of the studies focused around humans. Humans are horrible subjects for genetics experiments. Generally speaking, humans really don't like to be told who to mate with, and so on. So you have no control over who is mating with whom, which you really do need with genetics experiments. You need to know the parental traits and cross them with each other. You take a long time to have offspring to see how the babies are going to turn out, what traits the offspring have. It takes almost a year, 9 months, so that's really unwieldy. And usually these studies were limited to just a few little family groups. They couldn't be very broad studies. So Gregor Mendel did things differently. Gregor Mendel was a monk. He worked in a abbey, monastery that is in present-day Austria, and he worked crossing lots and lots of pea plants, among other things. He worked with the monastery where he worked was not. It was not unusual for the monks to be doing science. It was very much educated group of men that worked there, and they were all interested in scientific ideas. But Mendel was the only one doing the kinds of things he was doing, studying this intensely, the breeding of these pea plants. There were other monks that were interested in breeding of plants for sure, but he took it to a whole new level. This picture is showing what his garden area actually looked like at his abbey. This is showing what it looks like today. They still do grow plants in that garden in memory of him. You can see a little historical marker here. So what did he do that was different aside from not studying beauty being passed in humans? He did study pea plants. Much better than people. Pea plants are really easy to grow, and they are small, so you can grow lots of them in a small confined area like what he had. They're pretty quick to have a generation, so it doesn't take too much time to go from adult to seed back to adult again, so you can get your results pretty quickly. And pea plants also allowed Mendel to have great control over his experiments. And that is because pea plants have both male and female reproductive parts. They aren't just one or the other. They have both male and female reproductive organs, and these are hidden very well inside these petals. You can see these large petals here, where in these right here is where the reproductive organs are hidden. And that's really good, because what that means is under normal conditions, those reproductive parts are not exposed to the environment, and this plant normally will self-fertilize. It will reproduce by means of asexual reproduction. In other words, basically they clone themselves. That meant that if Mendel wanted to do crosses with these, he knew they weren't going to cross with any other plants unless he did it. He had perfect control over his crosses that he did, because normally they don't outcross with others. They self-fertilize. So, normally the sperm, which are carried inside of pollen grains, normally fertilize eggs within the same plant. The plants don't get random pollen from other plants that might be nearby, so that's good control. If Mendel wanted to breed two plants, then he knew he could just open up those petals and he could choose what pollen to apply to the plant. So let's understand a little bit about what he might have done. So here is a kind of cartoon pea plant. Okay, obviously this is a petal. Now these are I'm circling a male reproductive organ right there, and here is the female reproductive organ, which is basically this thing that looks like a vase in the middle of the plant. This thing that is female you might know as something called a pistil. The pistil has basically three parts. So if this is the pistil, it's like a vase, and it's open at this end. At the top, right there, or here, there is a place called the stigma, which is kind of a sticky place where the sperm are going to enter the female reproductive part. Okay? The bottom of the vase, excuse me, is called the ovary. And the ovary is where you will find eggs. Okay, so down here is the ovary. Inside of this structure in the ovary, you would find an egg. Okay, so this is the male reproductive part here, here, here. On this male reproductive part is where we produce sperm, but they're not just naked sperm. They are produced inside little tiny dust grains, if we saw one, called pollen. So there is a pollen grain, and inside it would be a sperm. So the pollen dust would land on, let me show you over here, would land on the stigma, and then a tube from the pollen grain would grow down into the ovary, and the sperm would simply swim through the tube and enter and fertilize that egg that's in the ovary. Okay, and then a little baby plant would start to grow, which we would see and call a seed. But this plant will undergo some changes when that happens. When the egg gets fertilized, of course, the petals will die away, the male parts will die away, and this female part, this pistil, will undergo some dramatic changes in how it looks. It will actually physically transform into what we would recognize as a pea pod. And inside would be many, because usually there's more than one egg in an ovary, and sperm will fertilize each of them, and so you would find these seeds that were created from each fertilization event inside that ovary. Okay, then what Mendel would do is just go plant those and see what kind of offspring he got. So how he would do these crosses then would be that he would get two plants, and let's say one would act as the mother and one would act as the father. So mom and dad plants. The first thing he would do before he did anything else was he would make darn sure these plants were what we call true breeding. And what that means is that he would let those plants self-fertilize. And he could let them self-fertilize 1,000,000 times. Obviously he wouldn't, but he could. And his goal was to ensure that every time those plants self-fertilize, they always produce the same type of offspring. For example, if he had a plant with pink flowers, here is his pink flowered plant, it would always make plants that had pink flowers if it self-fertilized. He didn't want any surprises. Again, this is all about experimental control. He wanted to know his plants were constant, that they always produce the same kind of plant if left to their own devices. Okay, so controlling variables, and he could do that very easily with pea plants because he knew they were going to be crossing out with other things. So if he watched a plant long enough and saw that it always produced one color of flower, for example, he knew he was ready to go. So he's got his pure breeding plants. He would open up those petals where the reproductive organs are, and what we see right here are 3 stamens. You can see the little yellow anthers where the pollen would be produced, and essentially he would open up those petals and cut the male parts off. And essentially, if he had, let's say he had a pink flowered plant and a white flowered plant. Okay, well, he would cut the male parts off the white plant. So cut these off, take them over, here's his pink plant over here, and he would sprinkle pollen onto the female, onto the ovary on this plant. He would cut off the male parts, he'd cut those off of the pink plant, and he would sprinkle the pollen onto the female part of this white flower. And so thus he's removed the male parts from both of them, and he has crossed both of them. So now this white plant would have pollen from the pink flowered plant, and this pink plant would have pollen from the white flower plant on its ovary. And then he'd close the little petals back up, hide the little ovary and stigma from the environment, and then he'd just wait and see what happened. He'd wait for the seeds to form, and then once the seeds form, he would have to plant them and then see what color flowers did he get. Okay? So pea plants were a really, really good choice for lots and lots and lots of reasons. First of all, they have many traits that are either on or off, and there is no in between. Okay? There is absolutely no blending of traits with some of these traits. In other words, in pea plants, you either have tall plants or short plants, and nothing in between, no medium plants. Okay? And what this did for Mendel was it allowed him to figure out his results much more easily. There are some traits that do appear blended, and if he had studied plants with those kinds of traits, it would really have been hard for him to say, oh, blending's wrong, because it would have appeared right. These kinds of traits where you have one or the other and there's no in between made it much easier for him to discern the laws of heredity that we know he did. He examined heredity in seven different traits in pea plants. He studied height, flower color, pea shape, round or wrinkly, pea color. They can be either green or yellow, and a few others. And what we are seeing here is a picture of some of the many traits he looked at. We will talk mainly about flower color and height and things like that, but there were several yellow seeds versus green and so on. Okay? Now, Mendel did some other things that were also a vast improvement over what his contemporaries were doing. He used a true experimental method, not just some, oh, let's look at beauty in a family. He used quantitative data collection. He didn't just describe, oh, well, this person was passing on this trait or what. He counted his results. He had piles of notebooks full of data, counting how many pink flowered plants did he get, how many white flowered plants did he get, how many green peas, how many yellow peas, and so on. He counted pea plants that resulted from his crosses, and he analyzed that data using statistics, and this gave him incredible insight into what he was seeing that he could never have gotten any other way. He crossed thousands of plants. Remember what the people at his time were doing was looking at a few humans in a family or two. He was looking at thousands of plants. Thousands. Why? Again, multiple trials, real significant information coming up from all of these plants that he was crossing. Okay? So, a typical cross, as done by Mendel, would look like this. He would have two parents that differed in one trait only. Why just one? Why not 2, 3, 4, 5? Why? Because it'd be confusing if he did that. He's reducing his number of variables. Okay? He would call such a cross, we would call such a cross a monohybrid cross. Hybrid refers to a cross. Mono means one. So we're going to differ in one trait only. Okay? All right. For example, let's look at plant height. Okay? I'm sorry, hybrid I have as meaning difference here. We can certainly refer to it as that as well. So let's look at plant height. He would get one parent that was tall and one parent that was short. And all the other characteristics about those two plants would be the same. They'd have the same flower color, the same pea color, everything about them would be the same. Okay? What we are describing when we describe what a plant or anything else that is living looks like is we are describing its phenotype. This is one of those vocabulary words you need, and it's the term for the physical appearance of a trait in an organism. If you have blonde hair, that is your phenotype for hair color. What's your phenotype? Blonde. If you are tall, your phenotype for height is tall. It's what the trait looks like. The physical, remember, pheno and physical appearance. So the phenotype of a tall plant is tall, okay? Phenotype of a short plant is short. Pretty simple, okay? So a typical cross done by Mendel would start with those true breeding parents. Tall parent always has tall offspring. Short parent always has short offspring, okay? If they are left to self-fertilize. Okay. Now, he would cross the two parents, and remember, we're going to call the generation of the parents the P generation. That's our notation. So P generation is the parental generation, true breeding parents, and that's important that we remember that that's how they started. Okay. Now, he would cross those pure breeding parents and wait for their offspring to be born and plant them and see how they looked. This offspring is called the F1 generation. Okay? And what he noticed was that the F1 generation of his crosses always turned out all tall. So let's get this straight. He's crossing a tall parent with a short parent, and all of the offspring are turning out tall. So this is the P, this is the F1, okay? And they're all tall. Well, he thought, that's odd. What happened to the short? Where's the short trait? So he needed to figure that out. So his next step was to cross F1 plants with whom? Why, themselves. He crossed them with themselves. He actually would allow the F1s to self-fertilize, but it is the same as actually asking an F1 plant to cross with another F1 plant. We're going to cross siblings. I know, ooh, but they're pea plants, so get over it. So he waited, crossed them, waited, and then observed the offspring that would result from this cross of F1s. And what he would get was a generation that he called his F2 generation. And what he noticed was that when he did this, the F2 generation turned out like this. 3/4 of them turned out tall, but 1/4 were short. How could that be? We're getting the short trait back after it was completely gone for one generation. So the summary of his results then, in the F1 generation, one trait appeared to be lost. But in the F2 generation, the lost trait always reappeared, and that always did so in a particular ratio. Always appeared with 3/4 of the offspring having the unlost trait, and 1/4 having the trait that had been lost. So in this case, 3/4 would have been tall and 1/4 would have been short. So if he had 8 offspring, okay, then 6 of them would have been tall and 2 of them would have been short in that F2 generation. Okay, that is a 3-to-1 ratio, and it always turned out that way. Okay, so Mendel's next job then was to try to explain this kind of odd result. Here's what he said. He said that every trait that an organism has is actually determined by two factors possessed by an organism. So every single trait has in an organism 2 factors that determine it, what it's going to be. Today, we call these things that he called factors, we call them genes. He didn't have that word back then. That's what we call them now. Now, where do these 2 factors come from? Where do these 2 genes come from for each trait? Well, the male parent contributes one of these factors or genes. The male parent contributes one for the trait. The female contributes the other one. So one gene comes from your mom, if you're a pea plant, for height, and the other gene comes from the father for height. Okay? These factors or genes, however, exist in alternate forms, meaning it's not all the same. We can have different forms of a trait. We can have tall or short. We call these alternate forms alleles. So the gene for pea plant height exists in 2 forms or 2 alleles. We have the tall allele and the short allele. Okay? In the case of height and many other cases that Mendel studied, the one form seemed to be stronger, or one allele seemed to be stronger than the other. I bet you already know this. The term for an allele that can mask another is dominant. The term for the allele that gets masked is recessive. So, what appears to be dominant in pea plant height? Tallness, because it seems that to be there more of the time than shortness. So, let's say we have a cross then, with a tall male and a short female. Okay? Got that? Male tall, female short. All right? Our genetic notation rules usually use the first letter of the dominant trait as what will symbolize the entire gene being studied. So the uppercase letter of that is going to be the dominant, the lowercase will be the recessive. So for example, for the plant height, for the trait of plant height, we are going to use the symbol T because that is the tallness is the dominant trait. So big T will be tall and little t will be short. Okay? And there you go. Each parent had two alleles, because remember Mendel said it takes two factors to determine a trait. So each parent has two. Mom has two, dad has two. And since Mendel started with true breeding plants, he said that those parents, those original true breeding parents, must have genetic makeups as follows. The male, who was tall, had to have both of his factors being big T, because he always bred tall. The female, since she always bred short, must have been little t. Both her factors were also the same. This is another vocab word you need. It's genotype. A genotype is simply the term we give to the genetic makeup of an organism for a particular trait. Okay, the letter combination. So if I ask you what's the phenotype of a tall plant, I want you to tell me tall. But if I want to know what's the genotype of a tall plant, well, the first thing you would have to say, well, is it pure breeding or true breeding? And if I said yes, you would say, well, its genotype is big T. Okay? If I said, what's the genotype of a short plant? You would say little t. If I said, what's the phenotype of a short plant? What would you say? You would say, why, the phenotype of a short plant is short. Mrs. Coleman, that's a silly question. All right? So, when these parents are crossed, each parent sends one of its alleles. Each parent is only able, even though they have two alleles, Dad had two big T's, but when he sends an allele to a baby, all he gets to send is one of them. He cannot send both. The process that assures that he only sends one is called meiosis. If you'll remember, in meiosis, we are dividing the parent's genetic material in half, and we are going to have sperm as a result of meiosis or eggs as a result of meiosis, depending if we're male or female, that each only have 1/2 the genetic material of the parent. So if a parent has two big T's, in its sperm, it's only going to have a big T. If a parent has two little t's in its cells and an egg, it will only have one little t, because we have cut the genetic material in half to make those sperm and eggs. Okay? So, when Mr. Sperm, carrying his big T, meets Ms. Egg carrying her little t, those alleles get combined in the offspring. Okay? And since the parents were true breeding, there's only one type of allele that each can send. The dad can only send big T's because that's all he's got. He's only got big T's. The mom can only send little t's because that's all she had. So if dad is big T, and mom was little t, and they can each only send one, dad gets to send a big T, mom gets to send a little t, there's baby. You have no choice. There is no choice for those offspring to be anything other than big T, little t. No choice. Now, what did Mendel do? He crossed those offspring together. He actually allowed them to self-fertilize, but it's the same difference. So he would cross F1 with F1, two that are exactly the same. Okay? Big T, little t, big T, little t. Okay? So this is a little bit different because now we got some different things going on here. We got two different alleles instead of two that are the same in each plant. So, thus each of these can send either a big T or a little t to their baby in their sperm or egg. Okay? So, there is a 50-50 shot that a big T could get sent to the baby, from this parent, or a little t could be sent. It's 50-50. We don't know which one is going to end up in the sperm. We don't know. Could be either one. Okay? There's the same chance over here, 50-50 shot, either one. Okay? There is no control over which allele gets sent to be fertilized in fertilization. It is completely random. Okay? That was an important insight. That means our results can be varied. You could get an offspring. So here is dad of the F1. Here is the mom F1. He could send a big T in his sperm to the offspring. And mom could also send a big T. There's a 50-50 shot that he sends that, she sends that. If they do, there's baby. Big T, big T. Okay? But, and of course that offspring would be tall. But also, you could get an offspring that gets a big T from dad. Here's dad, big T, little t. Here's mom, big T, little t. He could get a big T from the mother, but get the little t from the father, in which case the offspring would get big T, little t. Okay? Now, what would that offspring be? Phenotypically, it would be tall. Why? Because we already said the big T was dominant, and it masks little t, so this guy would be tall. However, there is an equally likely chance that if this is the dad and this is the mom, that mom could send little t over to the baby and dad could send the big T. Baby would still be big T, little t, but it would happen a different way. Okay? Still be tall. It would just happen a different way with different parents sending the alleles. Okay? The last possible result is that if this is dad and this is mom, mom could send little t, dad could send little t, baby ends up little t, little t. In this case, baby is short. Okay? So the offspring would be little t, and it would be short. Okay? If you had piles and piles of offspring, okay, the laws of probability, and when I say laws of probability, it's like if you flip a coin, probability says that half the time you'll get heads, half the time you get tails. Well, if you have lots of offspring, the laws of probability or chance say that 1 out of 4 of those offspring, there's a 1 out of 4 shot that they're going to be big T, big T. There's a 2 out of 4 shot that the offspring will be a big T, little t. Why? Because there's 2 ways to get that. There's only one way to get this. Dad sends a big T, mom sends a big T. That's it. But in this case, you could get the big T from mom or dad, the little t from mom or dad. There's 2 ways to get it. And there's a 1 out of 4 chance that it'll be little t, little t. Okay? Because of those combinations of alleles, we can see, well, let's go back for a second. What's this going to be? This would be a tall offspring. These would also be tall. This would be short. How many would be tall? 3. How many would be short? 1. So, we can say that 3/4 of these thousands of offspring would be tall, 1/4 of them would appear short, and that is exactly what Mendel observed. This is a 3-to-1 phenotypic ratio, meaning for every 1 short, he saw 3 tall. Okay? Now, so a few other terms we need. We need the term homozygous. Homo means same, so big T is an example of a homozygous genotype. Okay, that's an individual that has 2 alleles that are the same for a given trait. So there's another homozygous we've seen, and that was little t, little t. Okay, Heterozygous means that hetero is different, so heterozygous means our two alleles are different. Big T, little t is our heterozygous genotype. Okay? Um, so that is our stuff about Mendel. Now our next step is to do some examples. So thanks for watching. If you've got questions, please bring them to class with you. Okay? This is not in place of me talking with you in class. This is just to try to get us ahead of the game. So if you've got questions, circle them on your handout, bring them to class, and we'll talk about it. And we'll also have time to do a lot of practice. All right? Thanks for watching. Catch you in class.